Hello, in this video we will discuss about the target of metformin, the signaling pathway and the insulin signaling pathway. So first of all we will learn about how to insulin will work and the insulin signaling pathway. So let's begin to understand. This is the person which that is when eats the carbohydrates. It means it will be mastication to digest. So the mastication to digestion. After the digestion, absorption into the blood circulatory system and the assimilation. Assimilation means the food will be part of the body. And in this way here you can see the absorption into the blood circulatory system, the glucose directly from the gastrointestinal tract, and the increase of the glucose in the blood is known as hyperglycemia, which that will lead to stimulate the pancreas, the beta cell to stimulate to produce the insulin. When insulin will produce, will target to the different tissue, the several type of tissue, which that contain insulin receptor. And this insulin receptor when bound with the insulin will trigger the GLUT channel 4, which that will lead to influx of the glucose and the glycogen formation. Or the adipose tissue, if so the glycogen minor, but the adipose side contain lipids will be more. The triacylglycerol conversion is known as the lipogenesis. So in this way here is the glycogenesis is the muscle and liver will be occur, while the adipose tissue will develop in the peripheral part of the fat, and you can understand the adipocyte. So let's begin to understand the insulin resistance and related to the metformin we will learn about. So this is the insulin which that is triggered due to the glucose increase in the blood circulatory system, will dispose the glucose into the tissue. So here is the insulin resistance mean the receptor damage, which that will lead to hyperglycemia in the blood. So anyhow, here is the metformin is the initial. The metformin is to exert its primary and anti-diabetic action through the suppression of hepatic glucose production. So in this way here is the metformin contain hydrochloric acid, which that will lead to less absorption of the glucose into the digestive system. So let's begin to understand the insulin signaling pathway. So insulin is contain tyrosine receptor. When insulin will bind, the insulin receptor substrate will stimulate to phosphorylation of the PI3K, phosphoinositol 3 kinase, which that will which that will trigger to PIP2 phosphoinositide diphosphate, which that contain 2 phosphate group, will trigger and phosphorylation into the PIP3, and this PIP3 will convert into the diacylglycerol and the IP3, and this IP3 we will not understand the basically the phosphodependent kinase enzyme used to bind to IP3 pathway, which that will lead to function several, but we will learn just our view. So here is the AKT trigger the AKT phosphorylation of the AKT protein, which that will lead to GLUT4 transporter translocate. This GLUT4 transporter channel will be bound with the plasma membrane of the cell. After binding with the plasma membrane of the cell, it means the facilitate diffusion of the glucose will be occur. This facilitate diffusion due to the blood circulatory system when increase the glucose will lead to influx into the cell, and in this way the hyperglycemia will ultimately hypoglycemia. So that is the normal mechanism. But in the case of the insulin resistance mean the receptor will be damaged and the insulin will not bind correctly or bind not stability will not correct. So in this way the insulin will not bind, the triggering of the GLUT4 transporter channel will not bind with the plasma membrane of the cell. In this way the glucose will unable to move inside, so remain into the blood circulatory system, cause the hyperglycemia. So AKT will also inhibit the FOXO1. FOXO1 is responsible, the gene will produce protein for the gluconeogenesis formation, glycolysis, due to the production of several enzymes, and atrophy. Atrophy means the muscle loss. So in this way, the muscle will develop due to the insulin. So anabolic, remember. While on the other hand, the apoptosis is the basically the MTP, BAD, BAX, FKHR, and caspase lead to apoptosis will also inhibit. So this is the inhibition. On the other hand, here you can understand the glycogen synthase glycogen synthase kinase 3, which that is stimulated by the AKT for glycogen synthesis, and necrotic factor kappa B is used for the cell survival and risk pathway. Risk pathway we will discuss in another video, so remember. So will lead to glycogen into the muscle and liver. So let's begin to understand the mitochondria. So remember, the insulin is the anabolic hormone, but it will minor work for the synthesis of and growth, like growth hormone, because it is used for the glycogenesis, lipogenesis, etc. So here is the mitochondria contain inner mitochondrial matrix, outer mitochondrial matrix, inner and outer membrane. So in this way, the inner membrane we will discuss contain electron transport chain in the plasma membrane like structure. The mitochondrial membrane, bilayer of phospholipid, contain 4 complexes C1, 2, 3, 4 and ATP synthase. In this way, here is normal function. The NADH will convert into the NAD plus because NADH oxidase enzyme, and the FADH2 will convert, and in this way the electron flow while the proton will pump out in the outer matrix, and this is the electron transport chain will lead to pump out the proton outside, will lead to pH decrease in the outside while inside is the increase. So here you can see the hypoxia, which that is also will lead to convert the basically the hard oxygen will combine with the proton and electron to form a water. So remember this, when the metformin is the target to decrease the C1 complex proton pump out. In this way, here you can see the ATP synthase enzyme will lead to decrease due to the proton influx. So in this way, ATP decrease will lead to increase the AMP by the more usage of the ADP. So when decrease the ATP in the electron transport chain through a metformin or other process, and in this way lead to ATP decrease will lead to increase the AMP to convert the adenosine monophosphate kinase protein. This is the kinase protein, the family of kinase, and in this way the ATP which that will decrease, it means the AMP will trigger the AMPK and will lead to ATP synthesis and risk pathway. We will discuss in another video. So the other factors will lead to AMPK increase the ischemia, metformin and AMP and nutrients. The ischemia means the decrease of the oxygen, the hypoxia, as well as the ischemia means the basically the less oxygen to damage the cell. So the AMPK also inhibit the mTOR pathway, which that is the important for protein synthesis and also lead to oncogenesis. So the cancer inhibition through AMPK, and that is why the metformin is important to decrease the cancer causing gene activation. So here is the metformin is thought to exert its primary anti-diabetic action through the suppression of hepatic glucose production. So in this way, remember, the decrease of the ATP will lead to AMPK triggering, which that will lead to basically also the GLUT4 transporter translocation will also increase. So in this way, the AMPK will trigger the GLUT4 channel to bind with the plasma membrane like a insulin working. So when insulin will not work, so the metformin will work to influx of the glucose also for glycogenesis. So remember, the glycogenesis due to the AKT activation, which that will lead to glycogenesis, and the necrotic factor kappa B for cell survival and risk pathway. Remember that metformin is same like function, but this will also inhibit the mTOR pathway. So insulin will not work when we can use primarily the metformin for glucose using as a glycogen formation and the production of ATP, as well as other processes, and inhibit the apoptosis and gluconeogenesis and glycolysis, as well as atrophy. So that is why the metformin is very important for development of muscle and growth.